Hello everybody and welcome to today's webinar. My name is Melissa and I will be your host for today. So with that, I'm going to hand over to today's speaker, Baron, who is here to talk to us about the EBA and some of the new developments that they have going on.
Hi everyone, thank you Melissa for the great introduction. So this is going to be a webinar on the European Variation Archive, specifically speaking the variant submission and accessioning, so how to access data. And I'll also be going over the access to the dbSNP files. So that will be the bulk of the webinar at the moment, just because this is what we're focusing on at the moment, and the data requested from dbSNP from the previous dbSNP users, it does seem to be in high demand. So we just want to show you guys how we're going to handle the data, what we've been doing, and how the data will be released. So I will start off with just a short, just some bullet points just to show you the format of the webinar, the route we'll take as we go through. So again, as previously stated, the bulk of the webinar will be on this dbSNP data exchange and release, but I also wanted to take the opportunity to go through the actual European Variation Archive itself and provide a live demo of how to access data. From there, we'll move on to variant representation and submission. This includes representing variants in VCF format. That's the format we accept the variants in. We do seem to get there seems to be quite a bit of trouble with some submitters and the representation of variants. So hopefully this section will just provide a bit more insight into that. And then we'll move on to the bulk of the webinar, which is the dbSNP data exchange and release. And finally, I'll finish off by leaving you with some takeaway messages and some additional information you can use. This will include some useful tools, some useful tips you can use for your VCF variant representation, and some useful tools if you want to follow our dbSNP data exchange release. So I will start with the first slide of the webinar, which will be just the breakdown and overview of the European Variation Archive. You may have seen this slide before. I think it's a good slide to summarize all the services at the European Bioinformatics Institute as a whole. So as you see, we usually separate our teams into different sections depending on the molecular pathway, what they specialize in. So on the left, we have our literature and ontologies together, and then we have gene and genomes and variation, but then we've got gene, proteins and metabolites, protein sequences, molecular structures, anything to do with chemical biology, systems biology, and finally our cluster down here, which is the molecular archives cluster. And then here we have the European Nucleotide Archive, the European Variation Archive, which I'll highlight here, which is us, and the European Genome Phenome Archive. We wanted to make it as challenging as possible to remember all the names, but essentially we do archive different data. So the Nucleotide Archive does any sequence data. We do the variant data. The Genome Phenome Archive does classified human data. And we also have Array Express as well, which handles functional sequences, the functional genes, or functional sequences, sorry. So to give an overview of the European Variation Archive, the EVA, as you know, we store variants. So these can be from any species. They can be within or across populations, including subspecies. Along with the variants do come in through submissions. We attach the associated metadata along with the variants. So if you are looking at a variant, you'll be able to see which study it came from, the experiment type and analysis pipeline used to discover the variant within that study, and the samples the variant was actually discovered in. And finally, we also add annotation to the variants. So any variants that are archived and submitted to the EVA, we run the ensemble variant effect predictor just to give some sort of value to the variants within the EVA. One of the main sort of sections that the variant effect predictor gives would be on the functional consequence. It also gives the effect on the gene and transcripts which the variant may reside in. And we also use it to calculate population frequencies from the variant and the study metadata. And I don't want to spend too long on statistics of the European Variation Archive. I do believe this was updated quite recently, but we now have roughly just over 900 million variants currently browsable within the actual website, and these span across 63 different species. So what I'll do, I'll move on to directly onto the EBA website just to give you a bit of a live demo. So the best way to get there is EBI-EVA. And this will take you to the homepage of the European Variation Archive. So we have the subsequent header tabs at the top. Those is the submit data page, which gives you general information about data submission, the key stages, how to submit data. I don't want to delve too much into this page as I'll be covering it during the actual slides themselves, but this is the page you want to visit if you are planning to submit data to EVA. We generally have all the information associated with the submission on this page here. But then we have three ways of consuming data from the actual website. So we have the study browser, the variant browser, and you can also access the data computationally using the API. So I'll start with the study browser first off. So we find that the study browser is generally used after submission, and the submitter wants to check if their study's been archived, how it's represented on the website, if all the data is correct. Or we also get researchers who are doing comparative genomics. They want to search for a study that's been referenced in one of the publications they were looking at, saying that the data has been archived in the EVA. They want to take a look at the metadata associated with the study and just the study in general, just to see how it relates to their own research. So for that, we have a filter section on the left-hand side. So from here, you can filter from short genetic variants, so those less than around 50 base pairs, or you can get larger structural variants, sort of indels, anything above 50 base pairs we've put the filter to. You can search by text if you know the study, the name of the study you want to find, and you can also do it by genome or species. There's quite a few species to go down. And lastly, for the last filter section, you can do it by type. So if you are looking for sequences that just have exome sequencing for functional, you can filter by exome sequencing, or if you're looking at arrays, you can type by arrays. But these are the general filter fields we have for the study browser. So that will be the filter on the left-hand side. On the right-hand side, you'll be able to find the results from the query from what you filtered. We have the project IDs lined up here, along with the name, the genome they are associated with, the sample, the type, and these are all sections you saw in the filter. So if we were to select a study, for example, blood fluke, it's quite a popular study actually. So as you can see, you've clicked on the study ID, it takes you to the study page where you can find more information about the study. This includes the genome, the sample used, the taxonomy ID of the genome, the reference sequence used, the platform, and we also have these two link outs to download the raw data. We have the submitted files link, which is, this will be the raw file sent to us by the submitter. The only difference the raw files would be from what the submitter has and what we have is that we've run validation on the files to ensure that they are represented correctly, and they do indeed map back to the genome assembly used. So we run that validation suite. I'll provide the link out at the end of the webinar where you can download the validation suite onto your local machine, but that's the validation we essentially run. Just to ensure that all the variants submitted are indeed actual variants. And then the second link we have would be the browsable files link. The data between the two is the same. So if you do want to download data from a study, you can use either one. The only difference is that the browsable files link is after we've run the variant effect predictor on the variants. And then the variants are then browsable via the variant browser. We give this browsable files link just to show where you can download the file straight from our FTP. And lastly, many studies have their associated publication. So if you do find a study from the EVA directly, and you do want to investigate the associated publication, we have the publication link at the bottom here for quite a few studies. I just wanted to show you, if you were to click on the submitted files link, it would actually take you to the ENA. So as I said before, they usually do sequence data, but during the submission process, we broker the data to them. So there's no need to submit to both resources. So again, this will have the same metadata associated. And then you'll have all the files that are downloadable, the raw files that you can download and use with the from the associated study, sorry. So I think VCF1, VCF2, this particular study seems to have the data split across different VCF files. So that's the study browser. Again, it's usually when you're looking for a particular study of interest, maybe they've referenced that they have archived their study in the EVA, and then you'll come to the EVA and you can observe the study directly from here. But what we find is most useful is actually the variant browser, which allows you to browse variants, individual variants of interest. So we have the similar sort of schema to the study browser, where we have the left-hand side for filters. We have a bit more options for variants this time, just because the wonderful world of genetic variation has so many different options and corner cases. So you can do it by assembly. Well, in this case, we've actually done it by, instead of split by genome, split by species, we've actually split this by assembly version. So as you can see for human, we have the build 37, but build 38. So those variants, if you are going to search for them, they are actually separated by a genome version. You can search by, you can filter by variant ID, chromosomal location, ensemble gene symbol. So if you do know the ensemble gene symbol for example, the gene STAT1, you can simply just pop that in. It starts off at bracket 2, and you can simply just put in the gene name, and that will show you all the variants we have in the EVA associated with that particular gene. And then we have a few more options here that are not shown in the study browser, such as consequence type, minor allele frequency, protein substitution, and studies mapped to this assembly, other studies mapping to this assembly. So these are just additional options. The filter options are provided from the ensemble variant effect predictor. So that allows you to essentially filter some variants from what exactly you're looking for from the annotation. And same as the study browser, we then show the results on the right-hand side in this window here. So you can actually have more than 10 variants set, so you can put 100 for the window. I'm not going to do that just for this particular webinar. And then you can also export the variants that are provided in the results window, either as a comma-separated values file, or you can also do it in VCF. We've put the beta sign there just because we're implementing this for all species at the moment. It works for all species right now. However, for human build 37, we can't export the data as VCF at the moment, but we're slowly working on a fix for this. Just because that was the assembly we found had the most variants by far. If you select a variant and scroll further down, you can find some information from Ensemble about the variant. So this will include the annotation, the file the variant was submitted from. So again, you can find this in the, similar to the study browser where you have the files associated with the variant. This is the file that the variant would be submitted from, the genotype, and the population statistics. So for this particular study, so whole genome detection of sequences from 6 horses from diverse breeds. The genotype that was provided for this variant was the homozygous alternative, around 66%, heterozygous 16%, and homozygous reference, which was 16% as well. But it's also split up by the different studies. So this will be a different study, which was a EQTL, discovery and association. Whereas, as you can see, the genotypes for this study will be different from the ones provided from the previous study. So you can also click on population statistics, and again, this will provide you with the statistics split again by different studies. So for this particular variant, we had it submitted by 2 studies. So we have the statistics for one study and statistics for the other study. So that's a way to look at individual variants. You can also do it just before I leave this page. You can also go to the variant information page. So for each variant, we have these 3 links here. Well, this particular variant doesn't have the ensemble dbSNP equivalent. If you do click on the variant page for EVA, this will show you more information for this particular variant, as well as this header here saying that this variant has actually been merged into another. So if you do click on a variant that has been deprecated or merged, it will provide the link out to the variant that seems to be currently in use within research. So that's the information on the variant browser. The last way to consume data would be through the API, for those of you who are more familiar with computational methods. So I won't go too far into the API, but the benefit of using the API is that you can integrate it into different scripts, into different pipelines to ingest data through a level of automation, essentially. The information provided by the API is the same as the information provided by the study and variant browser, so don't feel like you're missing out if you're using one method as opposed to the other. But the API, again, the benefit is that it can be integrated into pipelines directly. And you can also do quite large searches for the benefit of the bandwidth of the webinar. I'm not going to click on this one because this one searches all variants in a segment region. So see, these are some examples of queries you can run. This query, for example, fetches all information on a particular variant if you specify the RS ID here. Or you can search all structural variation studies through here. Or this one's quite a small one, so you can search information on a particular study. So this will be the information that is provided by the study browser. And using the API, the information is then provided in a JSON format. So the information can be easily parsed and integrated into whichever pipeline you prefer. So as you can see, I'm using Beautiful Soup to just parse the information on the web page, and it gives me information in these chunks here. So you can see the description and analysis. Again, all the same information that is provided by the study browser, just via computational methods, accessing it through computational methods. So that's it for the actual consumption of data through the website. I'll go back to the slides as I just want to carry on and show you what we will be moving on to. So this was just another page describing the API. I showed you some examples, and there are some examples as shown on the API webpage, which we just visited, and the link will be here. But I'll put this at the end of the webinar. So we'll now move on to variant representation and submission of data to the EVA. So this is the general workflow, the general pipeline that we use for data submission. So we have the initial submitter. They fill in the submission metadata, which I'll go through in a moment. And they have their associated VCFs associated with their project and their research. They send this to us through the EVA help desk, where we then run the data validation stage. So this includes the format check. The format check ensures that the VCFs or the VCFs submitted to us do adhere to VCF specification. And it also checks that the samples in the VCF are the same as the samples provided in the metadata. And lastly, it checks if the reference alleles provided in the VCF are the same as the reference alleles from the reference sequence used. Once the data has passed the validation stage, we then broker them to 2 other databases at the EBI. So the sample data from the metadata, this then goes to biosamples, and then the information itself, the variant data, as you saw from the submitted data files, these are brokered to the ENA. And then lastly, the sample data and the variant data is then finally stored within the EVA variant warehouse, the EVA study warehouse. And from submission, we are then able to provide this project accession, this PRJEB number, which the journals may be requesting for publication. For this particular instance, the data validation, the validation suite is available on our GitHub page. I'll put a link out to that at the end of the webinar. But for the actual webinar itself, I'll focus on the submission metadata and VCF file side of things. So just to come out of the webinar slides again, the metadata can actually be downloaded from our submit data page. So if you just head down to the key stages, and you can find the metadata here. So I won't go through it because there's quite a few tabs you need to fill in, unfortunately. But there's only 5 tabs that need to be filled in. This is essentially just to provide us with all the metadata associated with your study. So the submitted details pretty much speak for themselves. The project is just to describe the project as a whole, the project title, description, the center the project was conducted in. And it also has the publications link outs where you can link your publication to the relevant data within the EBA website. The sample information is provided whether it's pre-registered, this means it's already submitted to biosamples, or if they are novel samples, once you enter the information here, this is how the information is then brokered to biosamples. The analysis tab, where you can provide multiple analysis associated with a single project. And finally, the files tab. We usually fill this in ourselves as we have all the names of the files sent over to us. And then that allows us to essentially bring all the metadata from this one file onto the EVA website itself. We do this via a series of Perl scripts, although Perl isn't everyone's favorite language in the team at the moment. But that essentially brings all the data across to the EVA, and it aids in the submission and brokering of data across to the ENA itself as well. So that's how you access the metadata. For the VCF, so part of our job in the EVA is maintaining official VCF specification. So I'll give you a quick run through of VCF specification. So this is what a valid VCF would look like. We have the header line and some information lines to provide more information on each variant. And then we have the format line to provide more information on the actual samples associated with the variant. So if I split that up, so I'll highlight the file format. So this line and just don't highlight both of them. So that line and the chromosome header line here are the only 2 lines that are essentially compulsory in the VCF. All the other lines are just to add whatever information is associated with the variants themselves, and just to add as much information on the VCF, on the variants, themselves as possible. So we have, again, as previously stated, I'll try to section it out for you. We have the info lines. So they provide information such as allele frequency, the read depth, the number of samples associated with the data. And I'll show you how those are represented in the variants, which we can see down here. We also have the filter lines, which are for read quality. But the most useful lines we find are the info lines and the format lines. The format lines then allow you to add information to each sample within your analysis individually. So they're mainly used for the genotype of each sample, the quality of the genotype, and the read depth. And you can see some examples down here in these sort of mock variants provided. So we have the chromosome that is associated with the variant, the position of the variant within the chromosome, the locus. If it has been assigned a previous RS ID or SSID, we'll get into more about RS and SSIDs further on in the webinar. The reference allele, the alternative allele, the read quality, if it's passed the filter, if you have put a filter on your analysis, and if it's passed or failed. And then we have the info lines. So the info keys are actually detailed in the header lines here. So for NS, we know that NS corresponds to number of samples with data. We can see for this variant, the number of samples with data in this instance was 3. For allele frequency, you can see that AF equals allele frequency. Then we can see that allele frequency for this particular variant was 0.017, or for the one above, 0.5. And that provides it for the variant as a whole. But then we also have the format lines, which the format, so this will be sample 1, sample 2, sample 3. And then we can see the GT corresponds to genotype. And then the genotype is provided here. So homozygous, heterozygous, homozygous alternative. And that can be done for each sample which can be provided in your VCF. As I previously stated during the validation stage, one of the validation steps we do take is to ensure that the sample names here in the VCF are the same as the sample names provided in the metadata, just to keep consistency with the data and ensure all the data in the EVA website is normalized and consistent across the whole database. So I'll put a link out to the VCF specification at the end of the webinar also. Do give it a read if you ever get stuck with the VCF. It's quite a detailed document. It attempts to sort of counter all the corner cases you may find with variants. A big one we find at the moment is representation of structural variants. They are represented a bit differently than SNPs. So we're continuing to work on that, and we are active contributors to VCF specification. So we'll carry on with the variant representation and submission. So after everything's been archived, everything's been submitted, you've provided your project accession to the journal for publication. What happens next? The next stages are the assigning of SSIDs and RS IDs to variants submitted to the EVA. So from the end of 2007, there's been a sort of agreement, this split between the dbSNP database and us. So dbSNP is now responsible for assigning SSIDs, which are unique identifiers to variants. They handle all the human data side of things. And we do anything that's non-human, any other species, we do the accessioning for non-human species. We accession the variant, every single variant provided to us, providing its non-human variants, and they are provided back to the submitter in a VCF file like this. Similar sort of schema as the general VCF specification. So you have the chromosome position ID, which is the assigned SSID, and then the reference and alternative files, the reference and alternative alleles, So going back to the SSIDs, what exactly is an SSID? Submitters usually want to reference them in their publication. It's quite good to reference, use them as identifiers during your publication. But there are some rules with SSIDs. So say if we have this initial submitted variant, so on chromosome 9, position 254, 3564, reference allele, alternative allele, and this was discovered on the Bos-Taurus assembly ARS UCD 1.2, and that's the GenBack accession for the assembly. This is submitted to the EVA and then assigned a theoretical SSID of 111. But what happens when the same sort of variant is provided by another submitter? In that case, it will be assigned a different SSID, even though they are the same variant. The easiest way to think about this is that we also remap the submitted variants to other genome builds, future genome builds. So for example, we can take this variant and remap it to a more up-to-date Bos-Taurus assembly. It's not more up-to-date, but it's a different Bos-Taurus assembly. But as new assemblies are coming out with higher quality, we are planning to remap these variants. So for this example, we've remapped it to this new assembly here for Bos-Taurus. But then you'll find that as it's a new assembly, there's new bases included, there's bases removed. The position of the variant will have changed. So in this instance, it's also 254. The initial submitted variant was on 245. And you can also see that the allele has changed. So the reference allele in this case would be T, and the alternative will be A. So in that case, they are the same variant, just remapped to a new assembly, but that will result in the variant also being assigned a different SSID. Similar in another case, if we are going to remap it to another assembly, so this one, I think this is quite a popular assembly used for cow species, the Bostosaurus UMD 3.1. Once it's remapped to that, as you can see, the position will have changed again. In this case, the reference allele has not necessarily changed, but that will result in a new SSID being assigned to this variant. So essentially, the SSID is a unique identifier for a single variant, regardless if the variant has been reported before. It will receive a unique SSID for that variant. So if you are going to reference your variants within your publication and your journal articles, it would be best to use the SSID as it's quite a unique identifier. But to add more value to the SSID and to essentially add more, well, try to keep consistency across accessioning of variants, SSIDs are then sequentially merged and clustered into relevant RS IDs, and that's where you get RS IDs from. So there may be multiple SSIDs clustered under a single RS ID, and that just goes to show, and it essentially aims to portray how the level of evidence of the variant from the RS ID. And we do then have some new rules for RS IDs as well. So dbSNP have done this for quite a while for non-human species. They've been assigning SSIDs and then clustering them into RS IDs. So our job now, what we've been doing since 2007, is to simply import all of the non-human variant data from dbSNP into the EVA. The variants will be available via the variant browser that we saw on the website. But there are some corner cases we found in the dbSNP, the backlog of data, where we essentially couldn't, well, what we've done is that we ran validation on the variants we are receiving from dbSNP just to ensure that they are still in use and they are valid variants. But there were some cases where the variants weren't in use or we couldn't find any evidence that they were variants. So we wanted to make this experiences as useful and intuitive as possible, but we also wanted to keep the commitment of only keeping high quality variants part of the EVA. So I'll go through the different types of files we're going to be using. So if you take a look at the top here, if you go in for an assembly, so this will be the format of the release files from dbSNP. So we'll have 5 files associated with one assembly. So for this example, I'll be using this. This is a GenBank accession for a dog assembly. Unfortunately, I can't remember which one, but it's just using it as an example. So this is the first file that we'll provide. This will be, we found this will be the most useful to researchers. And this will be the assembly name and then the current IDs in a VCF format. And these will be the current variants that are in use right now and the current variants that we deem as high quality from our validation stage. These variants will be browsable via the EVA variant browser, and the RS ID originally assigned to the variant has been validated and can be mapped to the assembly provided in the file name. The chromosome and contact name provided. So under the chromosome name, we recently sent around a survey just to find which sort of naming you would prefer in the chromosome name. And we think that just naming them by the chromosome name would be most useful. Initially, we had these GenBank accessions here, but we found that won't be as useful for researchers, especially if you're using various bioinformatic tools. They seem to be working more useful with these CHR01 sort of naming. But for that, we did want to include the GenBank accession. So in the header lines of the VCF, we've included what they correspond to. So for CHR36, you know the equivalent GenBank accession is CM blah 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 36.6. So that would be the first sort of file associated with it. Again, these will be the high quality variants, the ones that we found were validated. We can map back to an assembly, and the first ones we imported from dbSNP. Now, the second set of files will be, this will be split by species. So this will be canine, I can't remember the Latin name for the dog species now, it's canine something familiaris, and that will have the taxonomy ID. And then this file will be unmapped IDs.txt. This will be very different from the first one. So these will contain RS IDs that we couldn't find that they could be mapped against an assembly. So they were in dbSNP in the backend of dbSNP, but we attempted to map them back to an assembly to discover how they were found, but we were just unable to do that. Whether it was we couldn't find an assembly they were mapping back to, so we had the flanking sequencing, and I've sort of cut off the image here, but that will be the RS ID of the variant. Any SSIDs that are associated with the variant. We provide the sequencing in 5' and 3', and we provide the flanking sequencing on the end of the VCF for each variant. So again, a part of our survey to determine how useful and what kind of data you wanted was sort of, we were in two minds whether to include this data, as clearly it's not used. But we found that it's part of the dbSNP data that was provided for the release, so we have provided it anyway, essentially hopefully finding someone that may be able to find an assembly it's mapped to, but as for now we couldn't find anything that was mapped to, so we provided the flanking sequencing for the flanking sequences when possible. The third file will be the merge IDs. So again, we're just using this example for this assembly. The RS IDs, these are RS IDs that should not be used. However, they will be searchable on the EVA website. Reason being they shouldn't be used is that they have been merged into another RS ID. So they've been deprecated, they're not used anymore, and they've been merged into a current RS ID that is being used. If you were to search them on the EVA variant browser or homepage, if you go on the variant page, they will have a variant page associated with these variants, but the variant page again will have that banner I showed earlier, just stating which RID it's been merged into, and you can then link out to that RS ID just to determine, or that will be the one that's in use at the moment. The consensus with trying to include, thinking about whether to include this in the release was that it may be quite useful for those working with legacy assembly builds. We found that many people did request it regardless of if they aren't being used or if they've been deprecated into different RS IDs, but they have been now being included with the release files. So we do hope you'll find those useful as well. And the fourth set of files would be the deprecated IDs. So these are IDs that shouldn't be used as they were deprecated due to missing information. So slugging through the backend of dbSNP, we found that there were some variants that had some missing information. Obviously, dbSNP's been collecting variant information for many years. So there will be some variants that were in the backend of dbSNP that didn't have all the information available. We have included this as it was part of the dbSNP import. But again, ideally these variants won't probably be used as they had some information missing, such as the reference allele. I think some of them didn't actually have an alternative allele, didn't state whether it was a deletion or anything like that. So we've just included these in a separate file. And the last files that we'll provided are sort of a hybrid of the previous two. So these are RS IDs that shouldn't be used. So then they've been similar with the merged IDs here. They've been merged into a different RS ID, but that RS ID that they've been merged to was then also deprecated. So they shouldn't be used. I can't think of a scenario that you would use these RS IDs. But again, the general consensus was that this was part of the dataset and they should be included in the RS release. So to summarize the format of the files, we'll have the main file along with the assembly accession. and these will be the files that will be available on the EVA web browser themselves. So if you are looking into any of these variants, you can find them on the variant browser, which I showed earlier, but they'll also be provided in a separate file for each assembly build. We then have the unmapped IDs where we couldn't map it to a assembly. The merged ones where they've essentially been merged into another active RS ID. So that would be useful if you do find a RS ID that has been deprecated, but you find out there's an equivalent that is being used at the moment to represent the the variant. And then you have these two at the bottom here, which they probably won't be used as much as the previous files, but again, they are still part of the data that was associated with dbSNP. So we are coming up to the hour mark. So what I'll do is that I'll summarize everything that's been gone through in the webinar, and then provide you with some links for information on variant representation. I'll also provide the link for the validation suite, just in case you want to download it onto your local machine and run format validation and an assembly check on your VCF. It doesn't necessarily have to be used on VCFs that will be submitted to us. It could also be used on VCF you have just generally generated during your research, and you just want to ensure they are being represented correctly. So going through the webinar, we learned that the EVA, it provides a permanent archival and accessioning service for genetic variation from any species. The data can be consumed via the study browser, the variant browser, and Computational Methods can also use the API if you do want to integrate large bulk downloads of data into a pipeline. The variants can be accepted in the VCF format. So I'll show you the link at the end for some various tools. There's the validation suite. We also have a VCF template. I'll show you that quickly, actually. So on the help page of the EVA website, there's also this help page for submissions. And if you scroll further down, you have this section on how do I generate a VCF. This is quite a useful section. So again, the only two compulsory lines in a VCF are these lines. So you can download this spreadsheet to VCF file. And it just provides a template of the compulsory lines. So if you are just if you are doing research and you have a single variant to report, a single SNP, instead of slogging through thousands and, well, not thousands, but quite a few software that are used to download and generate VCF files, you can just put in your chromosome, put in your loci, the ID, if you have an ID for the variant, reference alternative, and the quality filter and information lines associated. So that will save you going through looking for software, especially if you're a wet lab scientist. Ideally, you don't want to spend more time looking through software. You can essentially just report these variants using this spreadsheet here. And in the spreadsheet, there's a couple of completed examples. This is quite a simple example. So the only added line will be this info line here, and that's used to just provide the allele frequency. So allele frequency 0.5, 0.2, and 0.3. And then we also have a second example with a bit more abundance of data, some more information, just in case you find that there is a corner case with your data and you need to provide it in a different way. So that's one of the tools. It's quite a useful tool that we call the EVA VCF template. So that can be used to just report single variants or multiple variants that don't go into the hundreds or thousands, and you just need to simply input them into the template and send them across to us. There is instructions on the website of how to convert the data from the Excel format into VCF. But if not, you can simply send it to the EVA help desk email at the bottom, and we'll do the conversion for you. And lastly, this is the bulk of what we've been working on, the dbSNP non-human species RS release. So if you are a previous user of dbSNP, we will be releasing these files in the coming months. And the current RS IDs, the current variants that are in use at the moment. they'll be available on the EVA website via the variant browser. And you can use those. They'll be the data imported from dbSNP. And anyone working with non-human species will be able to use those. So just to finish everything off, just wanted to say thank you. This is the team working behind the dbSNP and just the general EVA. So we have our project lead, Thomas, our team leader, Christina, who's actually left. So she's just left us to handle the rest of the EVA. So we have the 4 software developers and myself. And some additional information I wanted to leave you with. I'll move on to another slide at the end. So I'll just leave this here just in case you wanted to take a picture or jot down any useful links. The GitHub page will be where you can find the validation suite, which you can download onto your local machine. There is a version for Windows and another version for Linux. So do ensure you are downloading the correct version. But then that can be downloaded onto your local machine, run from the command line, and it will validate any VCF files you have, as well as any VCF files you need to check against an assembly. The help page, which I just detailed, you can find it again on the general EVA website, but I've put the direct link here. It does provide a bit more information on submission, including the VCF template, and it does provide some recommendations for some softwares that can be used to generate VCF from next generation sequencing data directly. We have the link out for the official VCF specification, which we co-maintain. So this would be quite useful if you're unfamiliar with the VCF. I try to go through as much as on the VCF, the variant call format as possible. Again, the different types and just the general variability in reporting variant data. There are many corner cases where you may find that you don't know how to represent it in VCF. Do take a look at the specification. It does, again, it's quite a long specification, but it tries to detail all the types and all the different corner cases of variant representation you may find. And lastly, on the EVA homepage, we have the EVA dbSNP import progress page. I didn't previously mention this, but this page is just used to detail if you are following along and you are waiting for a species from dbSNP to be available in the EVA. This would just detail you the progress and the status of that. So for instance, if you were waiting on vervet monkey data from that assembly, you see that the dbSNP import progress is 100%. It goes down quite a while until you get to some 0%. They're mainly from bacterial species, but we are working the import progress and the dbSNP import. Again, it is ongoing work, and we are constantly working on importing new and existing data from dbSNP as we go along. So I just wanted to finish up the webinar by saying thank you for listening. Again, I hope the VCF tools and the VCF specification we provided proves useful and provides fruitful during your research. And we also hope that the dbSNP files will be useful to you. I'll leave my email on the bottom of the next page. So before we do a bulk release of these RS release files from dbSNP, we are happy to provide you with a preview for any species you may be working with. If we have imported it, we can provide you with a preview of the files, just so you can see the format, which I've gone through previously, which I went through in the webinar, just to determine how useful the format of the files are for you, how applicable they are to your research, and how you find the format generally works for you. So I'll pass it over to Melissa, and thank you for listening, everyone.
Thanks very much, Baron. We do have time for some questions. If you do have a question for Baron, you can write that into the chat box, which you will see on the bottom right-hand side of your screen. I'm going to give you a couple of moments just to do that, but while you're writing your questions, just a reminder that this webinar is part of our regular Wednesday webinar series, and we have a number of other webinars on many of the different EBI resources coming up through September through December. And you can find more information about those on the EBI training pages. So let's just take a quick look to see if there's any questions. So no questions from the audience at the moment, but I have one question, if that's okay. So when people are submitting data, is there metadata standards or guidelines that they need to follow or think about?
Yeah, at the moment there is. Apologies, I forgot to mention that. I'll try to bring it up for you and show you a better example quickly. So in the actual metadata itself, the file, we have these info sheets. They're not compulsory to be filled out, but they do try to provide as much information on the types of data you can enter into each cell. Essentially, only these cell headers in bold are mandatory, but we do have these other cell headers that just can be used to provide as much information on the study as possible. Again, the info sheets will provide more information on the standards and what can be entered into each cell.
Okay, thank you. Are there any other questions for Baron this afternoon? Okay, so there don't seem to be any further questions this afternoon. So I'd just like to thank Baron very much for a great webinar, and thank you all for joining us. I hope to see you again in a future webinar. Thanks, and enjoy the rest of your day.
Take care.